In Lab 3, students are going to have an opportunity to use their microscope to look at different types of cells. With every lab that we do, you will always have a set of lab outcomes as well as a set of keywords that we want you to learn. The outcomes for this lab include identifying differences between prokaryotic cells and eukaryotic cells. We're going to talk about how those are different in just a moment. You're going to learn how to make your own slides. We call those wet mount slides. You'll also be observing slides that we have purchased. Those are going to be called prepared slides. We want you to be able to identify cells on the slide. So can you tell me that, for example, you're looking at a potato cell or you're looking at a amoeba? Not only do we want you to be able to tell us what the cell is that you're looking at, but we want to know, is it a prokaryotic cell or is it a eukaryotic cell? So sort of what classification does the cell fall into that you're looking at? If it's a eukaryotic cell, we're going to ask students to say if it's a plant cell or an animal cell or a protist. Okay, if it's a prokaryotic cell, the only prokaryotic cells that we're going to be looking at in this lab are bacteria. And then finally, we are going to learn to distinguish and identify some specific organelles in some eukaryotic cells like the nucleus or amyloplast. We want you to know what those organelles do, what their function is, and what would be found within that organelle. As I mentioned, here's your set of keywords. We want to, again, begin to learn these. These should be bolded or highlighted words throughout the lab. All right, before we get going, let's talk a little bit about cells. So cells are what we call the fundamental unit of life. They're going to display all those different characteristics, those 7 characteristics of life that you learn about in Chapter 1. The cells ultimately were first observed in the 1600s, and ultimately lots of research has been done, and it's really led to this thing that we call the cell theory. The cell theory basically states that all living things are composed of cells. Now, some living things may be composed of only one living cell, like bacteria, a single-celled living organism. Other species, like yourself, are made of billions and billions of cells. So again, when we talk about sort of organisms, we're not all exactly the same. We're going to have differences. And one of those fundamental differences is how many cells are you made of? Now, when it comes to the cells that you're going to be looking at in lab today, you're going to see that there are some pretty significant differences. However, before we start to look at those differences, I think it's important that we recognize that all cells do share similarities or commonalities, right? So all cells, although they may come in different sizes and shapes, they're all going to have what we call a plasma membrane. The plasma membrane is sort of the barrier that separates the outside of the cell from the materials inside the cell, okay, the plasma membrane. And all living cells also have genetic material. So for example, today I'm going to show you some cells that have a nucleus. In the nucleus, the genetic material that we find, for example, let's say with your eukaryotic cheek cell, or in this case, a cervical cell, that would be DNA, right? That's our genetic material. It's going to be ultimately very important in allowing us to make proteins that help our cells maintain homeostasis. Now, before we get into the differences between different cells, or we even talk a little bit about how you're going to make cells, I first want to talk to you a little bit about really the 2 major groups of cells, those being the prokaryotic cells and the eukaryotic cells. So all cells that we look at, all living cells in general, are going to fall into one of these groups. Now, as we look at these 2 different cells as examples of prokaryotic cells and eukaryotic cells, one of the things that we'll start with is that prokaryotic cells are going to lack a membrane-bound nucleus. Now, membrane-bound nuclei are going to contain genetic material like DNA. So in our eukaryotic cells, you're going to see there's a nucleus down here, and in that nucleus, again, let's say this is your eukaryotic animal cells, the cells that make you up, we expect to find that genetic material, that DNA in that nucleus. Now, our bacterial cell up here, they do not contain a nucleus. They do have what they call a nucleoid. That's a region. That is not a membrane-bound organelle. So that is a distinguishing feature between prokaryotes and eukaryotes. Eukaryotes have a membrane-bound nucleus where the DNA is enclosed. Our prokaryotic cells here, they have no membrane-bound organelles. However, they do have genetic material. Remember, a unifying characteristic that all cells have, right? They have a plasma membrane and they have genetic material. So not all cells, that genetic material will be enclosed within a nucleus. Prokaryotic cells, no nucleus. Eukaryotic cells have a nucleus. Other differences between these groups. Prokaryotic cells tend to be more simple. They tend to be smaller than eukaryotic cells. You're going to observe this. I think it will be very clear when you get in front of the microscope and you're looking at these different cells that there is a significant size difference between the cells that fall into this group versus the cells that fall into this group. Now, when we talk about eukaryotic cells, these organisms may be single-celled, colonial, or multicellular. So we'll talk about what that means in just a moment. But the key here is that when you look at a prokaryotic cell and a eukaryotic cell, we really need to be able to say which cells we're looking at in lab fall into each of these categories. So in this lab, the cells that you're going to look at that fall into the prokaryotic category are going to be things like cyanobacteria and lactobacillus. Those are types of bacteria. Those are going to be representatives of the domain bacteria. For our eukaryotic cells that we look at, we're actually going to look at 3 eukaryotic kingdoms. We're going to look at cells that fall into the protists, the animals, and the plants. We are not going to have any fungi represented in the lab that we look at. So again, if you are shown a cell, not only do we want you to be able to say, yes, that's a eukaryotic cell, or yes, that's a prokaryotic cell, but we want you to be able to say which group of cells is it for eukaryotes? Is it a plant cell? Is it an animal cell? Is it a protist? And then finally, we want you to be able to identify specifically what is the cell that you're looking at. So I'll give you some examples of that as we get into looking at the cells more closely over the next couple of slides. Now, when it comes to eukaryotic organisms, they can be very diverse. So for example, some eukaryotic organisms are single cell, made up of one single cell. Some are what we call colonial, meaning that they could live on their own. Okay, they could survive by themselves, but they function better as a group. And then the last group are what they are called multicellular. Multicellular organisms are like you and I. We're composed of multiple cells, billions of cells, and that those individual cells that make us up, they're unable to survive on their own. So you can't pluck off one of my muscle fibers, one of my muscle cells, and think that it's going to be able to survive all on its own. So as we move through the lab, you'll see that we do use these terms to describe the different eukaryotic cells that we're looking at. Now, I'm not going to go too much into how we sort of make a wet mount slide because you're going to get an opportunity to do this in lab. But I at least want to touch on how wet mount slides are different than prepared slides. And then we're going to close this video and I'll make a new video where I go through all of the different cells that you look at. So when it comes to the slides that you're going to be using during the lab, you're going to have what are called prepared slides. They look somewhat like you're seeing over here in this picture. So the slide is already prepared for you. They're going to come on sort of trays. They're typically metal trays. They're going to have a nice little label on them that tells you what you're looking at. And then usually there's this sort of centered spot, and you will see that it's going to have like an elevated, it's not going to be completely flat. That elevated region that you see is what we call the cover slide region of the slide. And it's been permanently mounted on the slide. These are slides that we purchased. You'll get to use them. Once you're done, you'll put them back. In lab, you are going to learn how to make your own slides. As you make your own slides, you will find that you need some specific materials in order to do that. So you'll be given glass slides. When we use these glass slides, we do want to be careful. It's glass, it will break. When you get the slide and you begin to make your wet mount, you're going to take some distilled water and you're going to put a couple drops on your slide. You will then put your sample usually in that distilled water. Now, when it comes to lactobacillus, it might come already in a solution, and in that case, you'll just be able to pipette that out. But if it's not liquid suspended already, you want to put a little water down so that your sample doesn't dry out. We're going to use the light microscope, and remember, the light is going to give off heat. Okay, it's an energy conversion thing, and it will dry the slide, and it'll dry your organism out. So you put a little bit of water on the slide. Then ultimately, you'll put your specimen down. Some specimens have natural pigment, natural kind of color. So for example, when you look at Elodea, you're just going to be able to put this square little piece of plastic on top called a cover slip, and then you're going to be able to look at the sample. You always want to put a cover slip down over the sample, over the organism, so that your microscope, your objectives aren't sitting right down in the solution. That would be very bad for the lenses of your microscope. Some of the organisms that you look at are actually going to have to be stained in order to provide contrast. You want to be very careful with the stains. It will stain your hands, it will stain your clothes. So I always tell students, be very, very careful. Oftentimes what we tell students to do is actually to put the cover slip down first. You put a little sort of dye on the side, you move the cover slip over a little bit, and then the dye will actually wick across, and it gives you a really nice little concentration gradient and provides different levels of staining and makes it a little bit easier to see things. So that's sort of your intro to the cell lab, how you're going to make slides, different types of slides. The next video we'll pick up with, we'll go through the different organisms that you're going to be looking at in this lab.